Hello and a warm welcome to our webinar today, which is about the freshly launched TME-Kit, and we're very happy that you joined us. So before I'm going to introduce our speakers, just a bit of housekeeping. So on your left-hand side, there's a control panel, which hopefully all of you can see, and there's a chat and a Q&A function. I asked you that for the chat, you only use that if you've got like sort of general questions like, will there be a recording? The answer is yes. So use that for that, or if you can't hear anything. And then the Q&A section, this is where you can ask your questions. So you might already send some questions while the speakers are talking, but there will be a Q&A session at the end of the talk where we'll go through all of your questions. So if you have them, please submit them in the Q&A feature. So now, before I introduce our speakers, I actually have one question to you. We would like to hear from you where you found us, where you heard about us, where you heard about this webinar. This is really interesting to us. Thank you. Okay, so while you're completing this, I'm going to introduce our speakers today. So first up, we'll have Tom. He is a senior scientist here. He completed his PhD at Harvard, where he worked on cell signaling and cell metabolism of lymphomas associated with Epstein-Barr virus, sorry. He continued his research at MDC, where he worked on lymphomas and the Epstein-Barr virus using transgenic mouse models. And then at Dynamic42, Tom is now heading the cancer team and working on the development of cancer spheroid on chip models. He'll be followed by Emily. She's originally from Freiburg and began her bachelor's degree in biotechnology in Jena in 2019. And in March 24, she completed her master's degree in pharmaceutical biotechnology. And then since April 24, she has been a PhD student here in the cancer team at Dynamic42, researching the human blood-brain barrier on chip. She works closely with the Brain Cancer Research Group at the University Hospital Jena Pediatric Clinic. So now, without further ado, I'd like to hand over to Tom.
Just a second. Wonderful. Thank you, Jule, for the introduction, and also a warm welcome from my side. As Jule said, I'm Tom, and I'm one of the senior scientists here. So initially, I give a brief introduction to the company and our organ-on-chip model so that you get some idea of the background, where this tumor microenvironment kit was built on. And then Amelie is going to give a good overview of how the tumor microenvironment kit works and how this is implemented into our chip. And then I'll show a few of the highlights of what you can actually do with this tumor microenvironment kit. So first of all, who are we? We are Dynamic42. We are a company spin-off of the University Clinic in Jena, Germany. So here, right, located in the center of Germany. It's a big biotech campus around. ZEISS has its headquarters here in Jena, Germany. And besides the University Clinic, there's also a university and a few big German research institutes. As a company, we are on one hand, doing products and applications. So we are selling our biochip to academic customers, mostly empowering them to do organ chip research in their own laboratory. We also do contract research. So this is actually most of my time, where we do different model setups in lung, liver, intestine, and then from my side, cancer, and then investigate tox or other things in these models. Last but not least, we also do training and offer this here in courses on site in Jena, but also worldwide, traveling then kind of as roadshows to really bring the organ-on-chip technology to the various labs that are interested. DynamicOrgan system is purchasable as a kit. So this is how it looks like. So really you get the plastics and all the biochips you need together with a pump, all supplied by us. And this then in the end is going into the incubator. So these biochips are different sizes and models. So you can have them for a single barrier or for double barriers or for the integration of spheroids. So there come a few different flavors. But today we'll see the 2-barrier chip mostly in place then for this tumor microenvironment kit. The chip per se is microscope slide format, so this would directly go also under microscope. It's always separated in these 2 different culture chambers, so a left and a right completely separated chamber for biological replicates or treatment and no treatment controls. Inside, you always have them different channels, which are all kind of connected to these on the outside so that you can fill them with cells or media, depending on what you need. Inside, as in many biochips or in many organ-on-chip systems, you have a PET porous membrane. So here we have one porous membrane separating then here the upper and the lower channel. On one side, you can, for example, have endothelial cells, kind of the normal setting to mimic a vasculature. And then on the other side, you usually have epithelial cells. In our case now, this will be tumor cells. And then these two cell types are then separated by a porous membrane. So the cells can talk to each other, but are still spatially separated. Because of the ports that are connected to the upper and the lower channel, you can always add a perfusion and different medias to both sides and then have individual perfusion of. Important for the Dynamic 42 system is that you're not dependent on a big proprietary platform where you have to put in these chips. So we work with peristaltic pumps, which we supply one model, but this can also be adapted to other models you might already have in lab. And then these chips are just pure plug system connected with tubing to that pump. And then you're already ready to go if you only have an incubator and the system purchased by Dynamic 42. Very important for us as scientists at Dynamic 42, but also certainly for the customers, is that our platform is really made to optimize the assay options you have in the lab. So first, this means you have a fairly big tissue size. So in our normal biochip here, the tissue size you're getting out is about a 48 well size of tissue. So there's a big, there's a lot of cells in there. And then always you have also access to all of the cells and to the supernatant. The supernatant you can access through the ports, or if you have a reservoir sitting there, you can have also direct access in the reservoir. You can use this for direct sampling, analysis for cytokines, compounds, or whatever you're looking for in the supernatant. Importantly, you have usually a circular perfusion, so cytokines are also enriched in the perfusion. At endpoint, you can always recover your tissue, so you can use a scalpel to cut out this PET membranes. You have direct access to your cells. You can either use this for IF stainings, like depicted here, but also for recovering the cells, for flow cytometry analysis, for viability assays, for RNA or Western blot. Usually Western blot, of course, just looking at a 48 well size is a little on the lower side. Because the chip is microscope slide format, you can always directly put this under microscope, look for immune cell traveling, live cell labeling, or other dyes that you can look at live microscopy. So I'm often looking, for example, at mitochondrial membrane potential, or tracking, for example, lysosomal dyes. Last but not least, because we have this barrier here, we can also look for enzymatic activity, for example, transport from one side to the other. So from endothelial to epithelial side transport or other transport or enzymatic activities. So this is kind of in a nutshell, the Dynamic 42 platform. And having this in mind and as a background, we now build on this to build this tumor microenvironment kit. So the idea here was to create a more complex tumor, but how we really were getting there and how this really looks like, Amelie is now going to take over.
Yes, so also a very warm welcome from my side. Having worked quite a bit with the TME-Kit, I'm really excited to introduce this to you now. The idea, as Tom already mentioned, was to just create a sort of mixture where we can have a tumor cells in the biochip, combining this with a dynamic vasculature, but also having the possibility to incorporate an immune component. So adding immune cells in there as well, and then creating also this fibrotic tumor microenvironment. And having all this in one package is kind of rationale behind the tumor microenvironment kit, which I want to show you now. Just a few words regarding the biochip. This is a slightly different one that Tom has been showing in previous slides. We established this tumor microenvironment kit in the 3-channel biochip, which has basically 3 vertically stacked channels on top of each other. So we have the top channel over here, which is then separated by a PET membrane from the middle channel. And then underneath this, we have a second PET membrane, which then has a bottom channel underneath. Each channel is accessible via 2 ports, so that's why we have 6 ports on each cavity of the chip. And the ports are designed as for all the other chips Dynamic42 has in a Lure plug format. So also each channel can be perfused individually if needed. The model setup, just to show you the empty biochip from a side view right now, we have a top channel, which is then separated by a porous membrane from the middle channel, and underneath the bottom channel with the second porous membrane. What we wanted to do is incorporate or introduce a hydrogel, including tumor cells and building a microvasculature in there. This will all be done in the middle channel. And then we wanted to perfuse media and also growth factors through the top and the bottom channel to just give then enough nutrition and also access to growth factors through the top and bottom channel. What we do is we have the DynamicOrgan TME matrix. We add endothelial cells in there. We add tumor spheroids or tumor cells in there. And we can also incorporate immune cells, which is then provided usually by the user. And what we then do provide mostly is the TME matrix components. And this is how you will then get it. This is nicely packed into one of these big packagings. You have growth factors, which you will find then in this bag. We have the matrix component, which are sealed individually. So you can have you can open one of these bags, do your experiments, while the rest of the kit stays closed on the side. And of course, you will also get all the other equipment you will need in addition to the 3-channel chip kit that you can also buy and use for TME. The 2 vials here on the bottom left are essentially the rehydration buffers. So the TME matrix components are in a lyophilized format. So you can just rehydrate easily everything you need before your experiments, so it stays nice and fresh. And this is then the components you will get. So this is just a schematic. Basically, this is everything that is inside the packaging I just showed you. And we have 5 vials with the matrix component A and 5 vials with the component B. We have 2 dilution buffers, which belong to each matrix component. And then, of course, we are providing the growth factors, which then you can, of course, rehydrate with water or methanol, depending on which one it is. The growth factors are for angiogenesis, and the plasticware is, of course, not showed here, but you will have the biochips, which you will need, and the plasticware that is included inside the kit. Just to introduce the workflow, how this will work, you essentially have a matrix component, which you will rehydrate with the dilution buffer. You will add growth factor medium, and then you will have spheroids and HUVECs, which then goes into the matrix component A, which essentially then makes solution C. You can also rehydrate then the second matrix component and mix this then in a 1-to-1 ratio, which then will start to polymerize. You will add this mixture with the cells and the tumor components already in the chip, middle channel of the chip. And as we already writing here, this is the polymerization starts quite quickly. So there is a sort of time crunch when you mix the 2 components, where you then essentially within the next 15 to 30 seconds would insert then this into the chip, and then it polymerizes nicely within the biochip chamber. As I already mentioned, the middle channel is then loaded with this DynamicOrgan matrix, and we plug this with reservoirs, as you can see here on the top. So these are the 2 ports going towards the middle channel, which we then close off. And then we create a perfusion through the top and the bottom channel. This is circular perfused. So the growth factors go through the top channel, are then directed in the bottom channel, and therefore evenly distributed from the top to the bottom. So we have a more or less regular angiogenesis throughout the gel. The reservoir on the top, which is here, serves as a bubble trap. So this is essentially as well a air bubble free perfusion, and we don't have any issues with air bubbles. This entire chip can then be connected to the pump, as Tom already introduced, and everything is placed in the incubator. And this is then how we would leave this experiment running for 7 days, and then change medium every day. And then in the end, after 6 to 7 days, you would have a vascularized tumor. I'll now hand over back to Tom, who will show you the really nice applications we have for the TME-Kit.
Thank you. Wonderful. So we've learned we're integrating these TME together with different cell types into that middle channel, then perfusing immune cells or perfusing media through the upper and the lower channel, and thereby starting to create a tumor in the middle. So what we're going to look at now is the different kind of compartments we put into this chip that you can later on then combine together to form your tumor microenvironment that you're interested in. So first I want to talk about a vasculature, or actually more a microvasculature. So the idea here is that we are taking that chip, and then for using the TME matrix we are providing, we are settling HUVEC endothelial cells in the middle, and then starting to perfuse the oncogenic factors we're also providing to create this microvascular network. What you can see in our chip is really that here also the perfusion matters dramatically. So if you do this for about 6 days, so usually it starts about 4 days after seeding, and then it continues to 8 days, we see that we create this wonderful tubular network inside of our biochip. Perfusion really matters. So if you do this statically, although you provide the same amount of growth factors and the same amount of media, they don't grow as well. But then in the perfused setting, you really see outgrowth of this nice microvascular network. Importantly, you can also stain this then in chip. So our chip, of course, microscope slide format. So you can directly fix this then in chip and stain in chip. And you can really see this wonderful 3D structure that it's creating. Importantly, if you do a cross-section of this, it's really perfusable, so it does create a lumen. So the lumen we are creating within the 6 to 7 days maybe is about 60 micrometers, so it's a decent size of lumen we are creating in there. And then we can also use this to perfuse microbeads through this. So we introduced microbeads in the upper channel, and then saw as we're slowly migrating through the middle channel to the lower. And here's a 3D depiction, which you really see where these microbeads are really nicely setting inside of the vasculature, not directly traveling through the gel. So including these HUVEC endothelial cells into our provided matrix, perfusing it with growth factors really creates within 7 days a perfusable oncogenic network. A second part, of course, of tumors are fibroblasts. We are mostly talking here about pancreatic tumors in the company because there's a big research interest here, but certainly there are also other tumors, but fibroblasts and some of the tumors are a really, really important part of it. So it was important for us to also mimic this. To look at fibroblasts, we were looking at fibrotic spheroids, so where we have tumor core surrounded by a fibrotic shell. So these come already as pre-assembled spheroids that we are creating outside in an ultra-low adhesion plate. And these are then embedded into our TME matrix inside of the gel, and then it works exactly the same way as we've seen before. So the system is perfused on both sides, and what we are seeing then is over time that these tumor spheroids are slowly growing. Actually, the tumor core is growing very, very slowly, but we are seeing this tumor fibrotic shell slowly growing over time inside of the biochip, really taking up then the environment of the tumor, creating a really fibrotic shell around the tumor, which is much bigger than what you could create an ultra-low adhesion plate, thereby really creating these barriers we're also seeing in vivo that impact immune cell traveling to the tumors, that impact drug trafficking to the tumors, and certainly has a big part of the tumor biology, many tumors, not only pancreatic tumors. Again, you can, of course, stain this in chip because it is microscope slide format, so this is fixed now. and then stained in chip. And we can really here see on the right, we see this spheroid sitting there, and then this fibrotic tumor microenvironment really spreading out several hundred micrometer into that chip, really creating a thick barrier around the tumor, much closer recapitulating the in vivo setting than usually the fibrotic barrier, which comes in the ultra-low adhesion plate, which really stops here just a few micrometer above the tumor core. So we can create a vasculature and we can create a fibrotic network. Of course, we can also combine this together, so bring fibroblasts or fibrotic tumors and the vasculature together in one tumor, in one chip. So this is then a setup. So we combine spheroid seeding together with HUVEC endothelial cells into that gel, perfuse this now with the androgenic factors, and then see what happens over time. And indeed, after a while, we see these tumors which are slowly starting to grow into that matrix, then interfacing with the tumor vasculature. So this is about 10 days growing, and then again, it's maybe nicer to see in an IF image where you can really nicely see how these areas now, with the fibrotic area here on the left and then the oncogenic area here on the right, how they're interfacing. And now we can, of course, start to investigate how the blood vessels are growing into that fibrotic area, whether they are still intact or whether they get leaky in that area. And then later on, also whether drugs or immune cells are using this route then to go into the tumor. So the combination now allows us to look for angiogenesis. We can also look for a fibrotic area. And now, of course, a third component that we wanted to look at is immune cells. So an initial setup that you can run is really just looking at how immune cells that are perfused through the vasculature are then integrating into the tumor. So again, here we are building tumor spheroids in chip and then starting to perfuse immune cells through that channel and then see whether they are starting to invade into that tumor. This is a depiction of immune cells traveling our biochip. Again, we can image live. So here we can see a live image as these immune cells are traveling over that upper channel through our biochip. You can really see it. This is 50 microliter per minute. They are traveling at quite a speed. So they really need to start to interact with this TME, really actively invade. So gravity is not just going to pull them down, but they're really going to rather move along the channel rather than invading. What we've seen for multiple cell types, and here I'm showing activated cell type as cells, we are seeing that they are starting to invade into the tissue here. We are looking at activated T cells, just activated with CD3, CD28 activation, and then looking 48 hours after starting the perfusion, we see that the cells start to invade. So here, this is, for example, a control sample where most of the cells here depicted in whitish are sitting on the membrane, but then here in this depth projection where the deeper your tissue, you're seeing that T cells are starting to invade into that tissue. Interestingly, in the same tumor, and this is mostly because we do PDAC research here mostly, we see very few invading T cells into that PDAC tumor, which is correlating to the immune cold environment that PDAC tumor is creating. So we really see here that activated T cells are excluded in that tumor microenvironment. Still, these few invading T cells we can characterize. Again, we can stain in chips, so you can start looking for these now inside of the chip, so we can see this invading T cells. And we do see a few of these T cells really interfacing directly in this tumor microenvironment. And this is one of the research projects that I'm doing now here with my team, is looking what these T cells are, how they are polarized, how they are activated, to kind of see how the PDAC tumor is really polarizing or selecting for special T cells to invade and not to be excluded from the tumor microenvironment. Last but not least, so we can now really combine fibrotic tumors with a vasculature, also with immune cells. We can now, of course, also see what immune cells are doing with the microvasculature. So the idea here is look in a vascular oncogenic network and start the perfusion of the immune cells and see whether the immune cells are really traveling through the chip. And indeed, they are. So if we add the immune cells to the upper channel and then start to perfuse them through the system, we start seeing them here just in a bright field image with a fluorescent component, where you can start seeing them going through these microvessels through the chip. You can, of course, again, also stain this then afterwards, fix the cells and stain, and then you find them in this oncogenic network. And sometimes, maybe here looking at one of these 3D depictions, most of the cells will end up in the blood vessels, which are kind of here spanning through the chip. But some of the cells are now extravasated, so really they move into the microenvironment around the tumor, or sorry, around the blood vessel. And now we can also characterize what kind of makes the immune cells capable to move or not move. And then certainly starting with the tumors in there will be very interesting to see how they now impact immune traveling into this tumor. So these are, in a nutshell, all the capabilities we've done so far with the tumor. So if tumor microenvironment setup. So to summarize this, we have created a very simple workflow for a scientist to adapt. So maybe the initial work, it maybe looks a little larger than it is. Basically, you really just have two components, mix them together. You mix the cells into this, and then you bring it into your chip. It gelatinizes in the chip, and then you have a wonderful setup of cells mixed together in there that can make an angiogenesis, make a fibrotic environment, and also attract or not attract immune cells. We can always perfuse this, so you always have the dynamic component to this, which is really interesting, especially if you're looking at calf and immune cell biology, because they sense this stress and strain stress we are bringing in with the dynamic culture. So this is also a big part of what we're looking here at, how these kind of mechanical forces are impacting the fibroblasts on the one side and shaping the matrix, and also, of course, the immune cells as they're invading. And then this is, of course, then in this combination, you can build these wonderful complex tumor models that combine all these different aspects of a real tumor microenvironment. And this being said, of course, we will be most interested to see what all you guys are going to be doing with this tumor microenvironment kit, with our chip and organ chip technology in general. And it will be interesting to see where this is gonna bring us. So with this, I hope we gave you a good introduction into the tumor microenvironment kit, that you got a good idea of what it is and how it can function in the Dynamic 42 biochips to really empower you to make a higher impact in your tumor microenvironment research. So this is our team. A lot of people participated on this. Certainly, Amelie was one part of that. So thank you, and thank you for listening.
Okay, so yeah, thank you so much, Tom and Amelie, for giving us a bit of an insight on the work you've done so far with the kit, and to explain the kit a bit more in depth to everyone than we've done so far. So just before we move into the Q&A section, I have one last poll, because obviously we would like to learn from you what you thought about this webinar. So while you're doing this, maybe Tom, if you can skip one slide for me. Because I've already seen the question in the Q&A section. We do have training available on this, so if you're now curious and you would like to learn how to actually use the TME-Kit on our biochip platform, then yes, there is an advanced course on cancer and immune cell integration. And so far we have 2 dates for this planned for next year, one sort of mid-May and the other one end of October. So if you are curious, then definitely pencil that into your calendar and let us know if you would like to book a slot. Okay, so now let's move into Q&A. So first questions actually came from Isa. She has a couple of them. So Tom and Amelie, I'm going to ask just a few and then get the next one later to you. So the first ones are, which growth factors did you use? What were the effects on the cancer cells? And is multiplex staining possible?
So maybe first for the growth factors, I cannot completely disclose what's in the growth factors, or I cannot disclose it. I mean, a big part, or is certainly a growth factor that you're all familiar with, but I'm not allowed to say. Generally, we see very little impact on the tumor cells so far. So if I grow my tumor spheroids, generally, the growth factors are fairly low dosed compared to other settings, because we have the perfusion that additionally gives an incentive for these endothelial cells to grow. But at this point, I cannot disclose. Our idea is always to kind of grow with and without to see whether there's any differences. And then certainly once you start to investigate your tumor model, to keep them out in the end. So I grow this for about 7 days with oncogenic factors to create this tumor microenvironment network. But then if I'm starting to now manipulate the system to bring in the immune immune cells or to bring in a treatment, this is also where I would fade out the growth factors as the oncogenic network is already established. The other thing is multiplex staining. Generally, you can stain whatever your imaging setup can do. Generally, in the chip, of course, you can only do one cocktail because it's one chip and it's all is connected. This being said, you can actually cut out the you can open the chip just as I showed earlier, and you can remove the gel. So you fix in the chip, so the gel is now fixed, and then you can remove the gel, and then of course you could cut the gel into multiple pieces and then put this into different multiplexing imaging setups. So this would clearly also work. I'm not so sure whether I missed one of the questions.
No, that's perfect. So two more from Isa. Have you tried immune cell invasion in organoids, and metastasis modeling is possible through vasculature? Question mark.
So first metastasis. I can always remember just the last question, and then you have to tell me the other question again. But anyway, so metastasis is of course a big point we are starting to look at. I mean, there is no reason not to think so that it should work. I haven't really looked at it yet. So the idea, of course, you have from, let's say, publications, you have together microvasculature and the tumor cells next to it. So there should be no reason to be able to look for trackable, maybe stained tumor cells, whether they are going into that microvasculature and then later we find them in the perfusion or not. We just haven't really looked at this yet. So at the moment, the focus was really looking at the immune cells as they're getting in or out of the system, and metastasis was not a part of it. But clearly the pore size of the chip is big enough so that cells can travel. I mean, tumor cell traveling per se, of course, I've seen, just not out of this microvasculature model or not. But this is something me and my team are starting to look at, and certainly something exciting we're going to be able to investigate. And then maybe Yuli, you have to remind me of the first question again.
That's all right. The question was just if you have tried immune cell invasion in organoids as well.
So I just don't have access to real organoids here. So we usually mostly use cell line dependent models, because as a company, it's getting pretty tough for us to get my more primary organoid structures here going. Generally, of course, there's no difference to the setup that I was using. So you're creating a organoid spheroid structure inside and then looking how they invade. Of course, I've also done the other way around, not preforming the tumor spheroids in ultra-low adhesion plates and bringing them in, but really just seeding single cells in there and then making a tumor. You can also call that organoid, of course, is cell line based, so it's going to be very homogeneous. But anyway, so they were then grown inside of the chip, and then we were looking for immune invasion. So that clearly is also something that works. So you don't have to bring in a spheroid. It's just a little quicker to do all this, especially with the angiogenesis. And if you do single cells, some of the tumors really grow for much longer time than the angiogenesis would take.
Perfect. So we've got a question from Samir. He says, hi, thank you much for the great presentation. Thank you, welcome. The kit seems exciting. I have a couple of questions. One, will there be possible to get training? So I think we've answered that one. And the second one is the reagents and growth factors that are provided. Are they specific for pancreatic cancer, or can they be optimized for some other cancer types?
So generally, it's, I mean, I can disclose so much that it's a fibrin-based matrix and it's not optimized for PDAC. I intentionally kind of kept this out here. I do a lot of PDAC research and we are using this for this, but it's intentionally a fairly, let's call it vanilla matrix. So it's really something where we rather think the tumor cells and fibroblasts, especially, that we are providing and putting into the matrix, are shaping their own microenvironment. So we clearly see this for the PDAC, that especially with the stretch and strain stress, that the calves are growing into that tumor microenvironment, and the gel's just getting stiffer all around the tumor spheroid. So we see that they shape their own matrix around this, but the matrix per se is fairly blunt, so not really geared towards a certain tumor If you want to do this or need to do this, you can of course add a few of your own components to that, but I can promise you that you will still get the full gelling or full polymerization process then going. So of course you're free to improvise on that, but generally it's a very blunt matrix with the idea that the tumor cells themselves or the macrophages and calves polarize the matrix themselves.
Perfect. Another question, Sasha. Have you checked the effect on hypoxia with the TME vasculature and that reflect in vivo conditions?
We've not actually really not looked in that setting. What I can tell you is that generally we create a hypoxic core in these tumor spheroids, and we of course also create a hypoxic gradient in our chips, so that we know. So of course the vasculature up top, and now here in bottom when we keep it in circular, is providing the oxygen and the nutrients to the system, and there's always a gradient into these tissues. So we are familiar with this from our lung, liver, and other tissues that we are creating. So the idea would be that within that biochip you have already a certain gradient. Now with the microvasculature you would think that it's kind of a little just really focusing on the tumor spheroids, but as they're even in tissue culture, if you culture them in a normal tissue culture plate, I have a core, would think that they also have one in this kind of TME-Kit setup, but we haven't really looked yet. It's just one of the things we just have to stain this also with now one of these tracking dyes. I just haven't really gotten around to that.
Another question from Jana. How long can tumor spheroids survive in the chip?
So the PDAC tumors grow dramatically long in there. I mean, if you have a vasculature on there, usually your time frame is about, let's say, 10 days or so, maybe longer, but not over 14 days. So this is the vascular component to that if you add these endothelial cells. If you really just add the tumor and let it grow in there, creating the fibrotic environment, for example, or just tumor cells by itself, they can go very, very long. So I have breast cancer cells, which are growing to very big tumors within 30 days or so in there. Also other tumors which grow slowly can go for a very, very long time. Others, for example, squamous cell carcinomas grow quickly in there and it just overgrows the whole matrix, let's say within 10 days or so. So it's really a little dependent on your tumor type. But if you have one which is especially encased in the calves or tumors that don't grow that fast, they can go for a very, very long time. Generally speaking, they grow slower, a lot slower than they grow in a 96 well plate, even in spheroids, and not anywhere close to deculture, which is of course much closer to the reality of tumors growing. Tumors are not really growing by 100% every day, so it's probably much closer to the reality.
Thank you. And another question from Michelle. Did you also see your cells in the top or bottom chamber yet?
You don't really have to, as the setup is here at the moment. So usually the endothelial cells, we didn't really show this here yet, but usually these endothelial cells by themselves just grow out of over that time frame, they grow out of the gel and then create a monolayer up and above, which is still connected. So we can still perfuse everything, but they create this by themselves. You can probably speed this a little up if for some reason you need this endothelial barrier very, very quickly by adding endothelial cells to the top or the bottom channel by inverting the chip and then seeding it on the bottom channel. But usually this is not necessary. After this 7 days or so that they need to create the microvasculature, they will also use these two membranes as kind of a scaffold to create a monolayer.
Tom. And another one from Charlotte. How are you experiencing this with Western blots? Wouldn't the cell number be quite limited to get sufficient protein amounts?
So when I talked about Western blot, I was mostly talking about our general biochip with the 2-layer formats. Here you have 48-well formats. So for endothelial cells, it's dramatically lower the amount of protein you're getting out because you get such a very thin layer of endothelial cells. And for the other side, for the epithelial side, it really matters how thick your tissue is. For example, our gut tissue is really like a 3D structured, microvilli structure there, so there's no problem to do a Western blot with this. Here, you get quite a few spheroids into this. So we are talking about 40, 50 in that setting. So if you recover these spheroids afterwards, just digesting them and then getting them out, you can certainly also do a Western blot on this. So it's really dependent on what you're looking at. Of course, if you're then looking for invading immune cells or so, this is of course much less. When I was talking that it's not enough for Western blot, it was mostly because the endothelial barrier, for example, that is a 48 well size, but just very little protein in endothelial cells per square centimeter.
Hey, thanks, Tom. And then question from Sasha. I guess you can't say what hydrogel is being used, but could you describe what this stiffness is close to?
Yeah, so it is fibrin based, so that's all I can say, and we are really operating on a very low stiffness gel. So it's really optimized to do angiogenesis. I certainly am aware that for some tumors, you would like to have a more stiff matrix. Certainly if you're like looking for an invasion of immune cells or also interaction with some of the drug components or so. This is not really what we made it for. So it's really optimized for angiogenesis. And again, here the idea being, if we talk PDAC, for example, we do see a much stiffer microenvironment around the tumor after 10 days where it's just growing in there. So there's certainly a component to this. We are still investigating what exactly is laid down there. It's going to be collagen probably, but we will have to look at this, and then it's more your tumor shaping the environment and not so much dependent on what we put in there to begin with.
Thank you. And another one from Isa. How do you feed both immune cells and cancer cells together to keep them active? Do you need specific medium content?
So as we've done it at the moment, we usually keep them apart. So first bring in the tumor and the endothelial cells, and then later on bring in the immune cells. If you seed them together, of course, you will have to see how the tumor cells are interacting with your immune cells. Certainly, there will have to be some incentive for the immune cells to stay activated for such a long time if you want to do this for 10, 20 days, if you don't have patient-specific material also where you really would get like an activation through the tumor. So that's something that you would have to look at. The tumor in general stays happy in there, also with very low serum concentrations or so they stay very happy. For the immune cells, you will have to check. The only thing that I can tell you is once an immune cell is actually invading into the tumor or staying in there, they are usually much happier than what you're used into deculture. So of course, kind of coming from B-cell lymphomagenesis and mouse modeling, we did a lot of immune cell cultures for T cells and B cells, and this is nothing close to what you're used to from a 2D culture. So first, in the chip environment, they're anyway happier because they get slightly re-stimulated just through perfusion, right? They get a little stretch and strain, and then they are already surviving longer in perfusion. And once they get into that chip, right, they're sitting now in a 3D microenvironment with a lot of interaction partners, so they survive a lot longer in there than usual. What exactly is going to look like for your tumor? Now, certainly, we are mostly looking at this from PDAC, that you will have to see. We are certain, so kind of maybe one angle back, what we are mostly doing now is integrating macrophages or tumor-associated macrophages into that setting, and they survive for a very long time. But here, of course, macrophages also survive a little longer than T cells in a normal culture setting.
And then from Isa, is it possible to dissociate TME and isolate the cells and process for further omic analysis?
So that's a very good point. At the moment, we do have a recovery solution in the pipeline. It's not out there yet, so we can't give you a recovery solution at the moment. Generally, this is more because of supply issues at the moment than really functionality. So this is certainly coming soon. You can also digest it with trypsin, so that also works. So that I've done, but of course, it's a very crude process. If you're just interested in DNA, RNA, certainly that works. If you are rather interested also in surface, which we are with the immune cells, that the trypsin modifications may be a little harsh. Also, of course, if you start to use other medias for your perfusion then of immune cells, which include serum, then of course trypsin digestion is also a problem. The way we are, of course, creating the angiogenesis and also running the model mostly is then serum-free conditions, or really with yeah, let's say serum-free conditions. So then it's also possible to digest with trypsin.
And then I believe we're reaching our last question to also give Tom some well-deserved rest at some point. The last one from Marvel. Hello, concerning DNA methylation, to test epigenetic modifications, wouldn't the cell yield be scarce? Yeah, that's all right. Concerning DNA methylation, to test epigenetic modifications, wouldn't the cell yield be scarce?
Sorry, you can't hear me, but I didn't get the end of the what with the scarring?
No scars. So are there enough cells to do analysis of DNA methylation?
Honestly, I'm not so sure how much you need for DNA methylation. It's not a topic that I worked a lot with. Again, if you're looking for the tumor cells or so you can have a lot of tumor cells in there. So 40 tumor spheroids, a lot of cell material in the end. So I mean, compared to the normal biochip, right? 48 well site, we have about a 300 microliter tissue, so you can fit a lot of cells into 300 microliter. I mean, we know, right? If you pellet your cells, 300 microliters a lot. So the limitation here is compared to other organ chip models much lower because you have the 3 dimensions and not only the 2 dimensions. But again, I'm not so sure how much material you need for your analysis and which cell types you're specifically interested in.
Awesome. Thank you so much, Tom and Amelie. I really hope that for some people that attended, a lot of things are a lot clearer now. Should you still have questions, feel free to message us afterwards, and I will direct the question towards Tom. Delighted to take them. And you will also receive the recording of this webinar just a few days after the webinar has concluded. So thank you a lot for joining today, and I hope we hear from you in the future again. Goodbye.